All right, folks. So per the last screencast, we're going to spend some time now talking about these nitrogen bases, the one of the three parts of a nucleotide, which is the monomer of nucleic acids, and DNA is a nucleic acid. So we see on the screen here 5 molecules, right? Cytosine, thymine, uracil, adenine, and guanine. All of these are going to become very, very familiar molecules to us as we go through this unit. There are 4 of these nitrogen bases found in DNA, all right? That means any nucleotide in DNA can have one of these 4 nitrogen bases as one of their 3 parts. So they are adenine is A, guanine G, thymine T, and cytosine C. That note, even in the picture here, that uracil is only found in that molecule RNA, which we have yet to talk about. So you can separate them into 2 categories as they're separated here. You have the purines and the pyrimidines. Purines are double ring structures. Adenine and guanine are the purines. Then you have the pyrimidines, which are single ring structures. Cytosine and thymine are pyrimidines. Now, here's how I remember this. I promise you, this is how I do it. Pyrimidines is a longer word with a smaller structure. Purines is a smaller word with a bigger structure. That's how I remember it, folks. It's no more complicated than that. And that's how I would suggest you come up with something that works for you in the same way. So again, these are nitrogen bases, one of the 3 parts of a nucleotide. And again, nucleotides are monomers of nucleic acids, which DNA is a nucleic acid. So nucleotides are actually joined together in the following way. So if this is one nucleotide, okay, we have our nitrogen base, right, a purine with this double ring structure. We have the sugar, and then we have a phosphate. Now notice what's connecting this nucleotide to this nucleotide is a bond existing between the sugar and the phosphate, the sugar of this nucleotide and the phosphate of this one. So that's a nucleotide, like I said. The backbone of DNA, right, what holds it all together, right? I told you that one nucleotide is connected from the sugar group of that nucleotide to the phosphate group of another. It's actually just alternating sugar-phosphate groups. These groups bond together, and that's what holds the nucleotides together to make a nucleic acid. So there's our phosphate, there's our sugar, phosphate, sugar, phosphate, and sugar. And it just keeps going for millions and millions of nucleotides to be connected. Now, the nitrogen bases stick out sideways from the chain. And I don't really like describing it that way, saying sticking out. They actually point inward, right? As you can remember that DNA molecule that sits on my desk, those nitrogen bases point inward at each other, right? So the nucleotides on one side, nucleotides on the other, the nitrogen bases, this part here, point inward at each other into the molecule. So there's our nitrogen bases. Again, remember, pyrimidines, single structure, purines, double ring structure. So in the late 1940s and the early 50s, it wasn't understood how this molecule could carry genetic information, right? It was too simplistic. It didn't seem to do enough. And better yet, not only how could it carry all this genetic diversity information, but how was it going to put it into action as a phenotype? Well, Erwin Chargaff would provide the first piece of that puzzle. Erwin Chargaff was an American scientist in 1947. He discovered that the amount of guanine and cytosine are equal in any sample of DNA. So however much cytosine there was, right? So a nucleotide that has a nitrogen base cytosine, so that's what we're referring to. So as many cytosines as there was always an equal amount of guanine, right? And I should say, Erwin Chargaff really just provided the first hint of DNA's structure. It wouldn't be until we actually discovered the structure, which James Watson and Francis Crick did, that we'd have a better understanding of how this molecule is actually the chemistry of heredity. And then he found out, or he discovered, the same was true for the amount of adenine and thymine are equal in any sample of DNA. Again, remember, these are the nitrogen bases that are attached to the larger nucleotide. Remember, the nucleotide is a phosphate group, a sugar group, and then a nitrogen base. Cytosine, guanine, thymine, adenine are all nitrogen bases. They actually are the part of the nucleotide that make it different, right? A cytosine nucleotide is different than a guanine nucleotide, only because their nitrogen bases are different. So he discovered that adenine and thymine were always in equal proportions in DNA, and then he discovered cytosine and guanine were always in equal proportions in DNA. And this would become known as Chargaff's rule, that A always equal T and C always equal G. Now, the observation that A equals T and C equals G became known as Chargaff's rule, and at the time the observation made, it was not clear why this fact was so important, right? Because we didn't really have much of a structural idea for DNA yet, at least not one that was accurate. So this was like the first piece of the puzzle, but it was a piece that we'd need later down the road. So we didn't see it for what it was just yet. And then enter Rosalind Franklin. She was a British scientist. She was actually known as an X-ray crystallographer, and in the early 1950s, she actually started to study DNA herself. She used a process called X-ray diffraction, right? X-rays are wavelengths of light, and she was trying to visualize an actual DNA molecule using X-ray diffraction, trying to almost get a negative space image of it, right? Like shoot X-rays at a molecule of DNA with a backdrop to it, and then you get the outline of DNA on the backdrop. That was her goal. Now, she would do this with large purified samples of DNA. She'd aim powerful X-ray beams at the samples, and then she'd record the scattering pattern, like I just described. The scattering pattern is the outline on the backdrop behind the DNA sample that she's shooting X-rays at. Now, this image right here, this is the image that would shake the scientific community, at least those who were solely working on DNA, because this shows a clear pattern and symmetry and shape of DNA. Now, again, by itself, to Rosalind Franklin, who was actually not a DNA expert, she just was working on DNA. Watson and Crick were much more the specialists on this molecule. So to her, this X-ray didn't reveal much about the molecule. But it would provide clues to it. It could provide clues to its structure, but once Watson and Crick got a hold of it, they would discover the structure of DNA. Now, the X-ray does show, which you see here, that strands of DNA were twisted around each other in a shape that we would call a helix, right? Like a winding staircase, like you see the molecule on my desk. And the X-ray suggested that there were two strands in a structure of DNA. So DNA would be coined double-stranded. And it appeared that the nitrogen bases, as we can see, these kind of, the nucleotides here, these black lines, are kind of oblong and the longer end points inward. So it looks like the nitrogen bases were actually at the center of the molecule, oriented inward. And we later, have confirmation, thanks to Watson and Crick, that this was all correct. All right, folks, I'll catch you in the next.